Okay, so for this note set, you'll want your pictures, your diagrams out, and because I'm going to use the diagram some, and then you may also want paper kind of near the end. All right, so this is the same picture that you have on the left side of the diagram that we labeled in class. We labeled the neurolemma, the Schwann cell, the myelin sheath in class. So this is the one on the left side. So we're going to go ahead and label some parts here. The rest of the things that were on the structure list that we can talk about these parts and you'll know what's going on as we use these words. So starting here first with these branches down here, these shorter branches here, these are the dendrites. The dendrites are important for taking information to the neuron cell body. So this long one here, this big process here, that's our axon. And so the axon takes information away from the neuron cell body. But the dendrite is taking information into the neuron cell body. You can see the little black arrows are headed towards the neuron cell body, where on the axon they're headed away. So on that note, this part right here that looks like a cell, that is the cell body. You see the big, large purple nucleus in the middle. You'll see lots of mitochondria to make ATP. You'll see lots of these little things called Neasel bodies. Those are unique to nervous cells in particular. So the Neasel bodies, they help make some of the extra proteins and stuff that are required to conduct the action potential. So they're going to be specific to nerve cells only. So we've got the dendrites bringing information into the cell body, the axon taking information away from the neuron cell body. Okay, these here, just like on our other picture, right, we've got Schwann cells with myelin sheath. And in between the areas where there is no myelin, so these little areas in between here, those are called nodes of Ranvier. It's a French guy, so it's pronounced all fancy. So those are the nodes of Ranvier. So the nodes of Ranvier, they don't have any myelin. Myelin is an insulator. So just like in physics, okay, where you talked about insulators versus conductors, we are talking about electricity because we're talking about nerves. And so the impulse is an electrical impulse that is traveling down the neuron. And so the areas that are surrounded by myelin sheath, they are insulated. So no electricity travels through that area. But the nodes of Ranvier, which are also sometimes called internodes, I-N-T-E-R, between, right? So the nodes of Ranvier, which are also sometimes called internodes, okay, those are non-myelinated areas. So they do not have any myelin sheath. So the electrical impulses, the electrical message, can travel easily through these nodes of Ranvier. And so what will happen is we will end up with two types of conduction that is possible for the nerve cell to do. The nerve cell can have what's called continuous conduction. And so continuous conduction would be on nerves that have no myelin. So the action potential has to travel all the way down the neuron, touching every part of the neuron, depolarizing every bit of the neuron, of the axon. And then the other option would be what's called saltatory conduction. This is what you see happening on this particular picture that we have here. The action potential will jump from node to node all the way down the neuron. And if you think about it, that's going to be significantly faster than if it has to travel all the way down the neuron if there was no myelin. So the myelin sheath acts as an insulator so that electrical messages can't travel through that part. But by acting like an insulator, that will actually speed up conduction. Myelinated nerves messages travel faster on myelinated nerves than they do on unmyelinated nerves because, again, they can use this saltatory conduction. They can jump, they can take big leaps instead of bunches and bunches of little tiny steps to try to get from one end of the neuron to the other. In addition to the myelin, the diameter of the neuron, the diameter of the axon, also affects its ability to conduct messages. It's how fast it can conduct messages. The larger the diameter, or the faster the message will be conducted. So again, just because I'm not writing everything down, make sure you get it down. I know I'm repeating a lot, but that's to make sure you still get it written down. So myelin or no myelin impacts how fast the message can be conducted. Myelinated nerves are faster because they do saltatory conduction as opposed to continuous conduction, which is done by unmyelinated nerves. And then again, the diameter of the neuron also influences the speed of conduction. The fatter or the larger the diameter, the faster the current will be conducted down the axon. The myelinated fibers are called white fibers or white matter, and the unmyelinated fibers are gray matter. So white matter will conduct messages faster because it was myelinated. Gray matter will conduct messages a little bit slower. The myelin is made out of a lipid or a fat. The myelin is made out of fats, and if you think about it, if you've cooked meat at your house and you've let the pan cool, that grease will then turn white as it cools and becomes solid at room temperature, hence the white matter for the myelinated neurons, because they're wrapped in this insulating fat. Okay, and then down here at the end of the neuron, we saw these when we did muscles. We have the little axon terminals. They are part of the synaptic knob, and these are our axon terminals. So this is the very end. So down here at the end here, this neuron is going to come in contact with a muscle cell, a gland cell, another neuron. Okay, and they're going to contain, this is down here at the end, is where we're going to contain all those neurotransmitters. So we're going to look at a synapse again, and it's going to be very, very similar to what it was with the muscle. So here's our synapse again. You guys have this diagram as well. It's not labeled for you, but you should hopefully know what most of this stuff is. So I'm down here at my axon terminal, right? This is the end of my axon. I'm at the axon terminal, and the enlargement at the end is sometimes called the synaptic knob. There's all my synaptic vesicles. Remember, those synaptic vesicles contain neurotransmitters. Depending upon what the nerve is trying to talk to, it will depend on what neurotransmitter it is. Remember, when the nerve is talking to a muscle, it's acetylcholine. It could be serotonin. It could be dopamine. It could be GABA. But those synaptic vesicles are going to contain the neurotransmitters. Here's my presynaptic membrane, which is on the end of the neuron, and the postsynaptic membrane, which is on the cell that is receiving the message. Remember, our synaptic cleft, or our space in between, is full of interstitial fluid. So again, this all should look very, very familiar to you here. Nothing is different here. This is all the same. We're just not using the specific muscle example here. So I have a postsynaptic membrane. I have generic neurotransmitters here. So let's look at how this message is going to travel down the neuron, down the axon, away from the cell body, down to the end of the axon. And so this graph should look a little bit familiar. We talked about this when we talked about a muscle cell depolarizing, and we're going to add a little bit to this. So I'm going to kind of break apart this graph. You don't have a picture of this graph, but you could sketch it. You could hit pause real quick and sketch it. Okay. What's on the left here is our membrane potential. Remember, our membrane potential is the difference between the outside of the cell membrane. Let's say this is the outside and this is the inside of the cell. And at rest, we'll see right here, our cell is negative, meaning it's more negative on the outside, or on the inside, and more positive on the outside. It's more negative on the inside due to ions and proteins. Proteins have big giant molecules with big negative charges. On the outside, I've got a lot of ions that are positive. Now remember, just because it's more negative on the inside doesn't mean I have no ions on the inside that are positive. It's just more negative on the inside. So at rest, this membrane is considered polarized. Remember, the membrane is polarized at rest because it has a charge, right? So again, this is the same as muscles. So it's polarized at rest because it has a charge. And I'm going to use this line right here to be my membrane, right? And what we see happening on when the neuron is at rest is I've got a lot of potassium on the inside and a lot of sodium on the outside. Okay? A lot of potassium, these proteins on the inside, so it makes it an overall negative and an overall positive with all that sodium on the outside. So this is what it looks like at rest. This is what it's polarized, and it has its resting potential. And it will become depolarized when those sodium channels open. So when the neuron receives a message that is sufficient enough to open up those sodium channels, so it's just like the muscle cell, sodium channels will open, right? So this is what it looks like at rest here. And then when it receives a strong enough stimulus, those sodium channels open. I left out the word channel, but I don't have a lot of room here. So the sodium channels open. So when the sodium channels open, just like in the muscle again, sodium goes inside. And so if sodium goes inside, if you come back to your graph, right, I'm going to start to head up towards 0 because I've got a bunch of positives coming inside to where my negatives are. So I'm heading back up towards 0, and that process is called depolarization. So that's happening when sodium is entering the neuron. So sodium's entering the neuron, that will depolarize it. You'll notice we passed a little line here called threshold. Threshold is the amount of depolarization that needs to happen to cause an action potential, to make that neuron conduct a message. Okay? If enough sodium has entered to change the voltage where it reaches threshold, then an action potential will occur. And just like muscles, neurons have an all or none situation. If enough stimulus occurs for the neuron to reach threshold, then an action potential will happen. Okay? If it doesn't reach threshold, then no action potential. There's no like medium action potential or a little tiny one because it barely reached threshold. It reaches threshold, it gets a big, full, giant action potential. It doesn't reach threshold, it doesn't get an action potential. So there needs to be enough stimulus happening to make enough sodium channels open so that I get enough depolarization to reach threshold. I reach threshold, all or none kicks in, and I get an action potential generating on that neuron. Well, once that action potential is generated on that neuron then also needs to repolarize. It's got to reset. If it doesn't reset, then it cannot do a new action potential. So it has to reset back to its resting membrane potential, which means it needs to go back to being more negative on the inside. It needs to repolarize itself. And so to do that, potassium is going to leave the cell. And this will work because just like sodium, potassium has a positive charge. So potassium will leave the cell, and that will electrically reset the cell. The cell will go back to being more negative on the inside and more positive on the outside. The problem is the ions are in the wrong place. I've got sodium on the inside now with the proteins and potassium on the outside. And so eventually the neuron is going to have to completely reset that. Okay? And so the neuron will repolarize. So it's headed back down. Potassium is leaving. That's what's causing this repolarization. And it will hit what's called a refractory period. And during that refractory period, that neuron cannot conduct another action potential. It doesn't matter how strong the stimulus is, that neuron absolutely 100% cannot conduct another action potential. And because it cannot conduct another action potential, that you can think of it as like that neuron is kind of partially frozen right there. So it cannot conduct another action potential during that refractory period, because what it's doing during that time is it's got to get rid of the sodiums. It's got to make the sodiums and the potassiums switch places, right? It repolarized and it reset electrically, but I've got to get the potassium back on the inside and the sodium back on the outside. And that is going to use the sodium-potassium pump. And so your neuron will actively pump the sodium out and the potassium back in. So it's active, so that's going to require energy. It's going to require some ATP, and it's going to physically push that sodium out and then pull that potassium back in, so that the neuron will eventually look like it did before, with sodium on the outside, potassium and proteins on the inside. And so these are the steps that the neuron will go through as it forms the action potential. So it started off more negative on the inside. It got stimulus to open up sodium channels and got enough to reach threshold. So it depolarized enough to reach threshold, so it created an action potential. Sodium, lots of sodium rushed in. So now it has to reset now that it created the action potential. So the potassium will leave. It'll go through repolarization. Okay? It'll hyperpolarize, so it'll go past threshold. It'll hyperpolarize, so it can go past threshold. And during that time, it cannot create any new action potentials because it's resetting, it's pumping that sodium out and the potassium back in. Okay, so again, very, very similar to muscles. And so we will do an activity with this in class and model this in class. So we're going to look at two more things really quickly. We're going to look at ways to classify neurons. If you remember with joints, we did functional classifications of the joint and we did structural classifications. So we classified the joint by what it could do, and then we classified the joint by how it was built. So we're going to do the same thing with nerves. So the functional classification of our nerves, what they can do, we've already talked about this. Okay? We basically have three types of nerves here. We have afferent neurons. Oh, let me get a color you can see better. So we have afferent neurons. Remember, afferent neurons are sensory neurons. My touch screen isn't working very well, so we're going to speed this up. So we have afferent or sensory neurons. We have efferent, which are motor neurons, so carrying messages away from the central nervous system. Afferent carries sensory information towards the central nervous system. Efferent carries it away. And then we have what are called association or interneurons. Okay, remember, inter is between. So association or interneurons, these are carrying messages between neurons. So we're going to find these actually in the central nervous system. So where afferent and efferent, carrying those messages to and from the central nervous system, are located outside of the central nervous system, these are carrying messages between neurons, so they are inside the central nervous system. We can also classify nerves structurally, and we're going to look at three classifications of nerves structurally. So the first one is what's called a bipolar neuron, right? Bi is two, meaning it has two processes off of the cell body. So one main dendrite, one main axon. So two processes off of the cell body. Again, one main dendrite and one main axon. We're going to find these associated more with our special senses, so in the eyes and the nose. Another one is a unipolar neuron. So with a unipolar neuron, I have one axon. You'll notice the axon is referring to both processes there. So the cell body branches off, one process branches off the cell body right there. So one axon branching off of the cell body. And then dendrites will just be at one end of that axon. Okay? These are most of our sensory nerves in the peripheral nervous system. And then our last one is what we think of as a typical neuron, a multipolar neuron. So a multipolar neuron has one axon, but the multi part is the dendrites. Lots and lots of dendrites. One axon, multiple dendrites. So all three types of neurons have one axon. It's the dendrites that are a little bit different. Bipolar has one dendrite. Unipolar just has the axon coming off the cell body and then multiple dendrites at the end of the axon. And then a multipolar neuron has lots of dendrites coming off the cell body and just the one axon. These are going to consist of all of our motor neurons are going to be multipolar neurons, as well as all of those interneurons or association neurons. Interneurons are also called association neurons. So structurally, we're going to classify it by the branches that come off of the cell body, and then functionally by the type of message that it can carry. Okay, so that is the bulk of our nervous system information. Like I said, it's repetitive on muscle system. We are going to look at reflexes together in class, and we'll model some of this action potential stuff. We will hopefully get to go outside and model some of this stuff, and we will hopefully get to do a lab modeling reflexes as well.